Episode 3, The Genetic Influences in Psychopathology. Introduction, Unraveling the DNA of Mental Health. Welcome back to Mind Diagnosis Lab. Today we embark on a deep exploration of one of the most captivating and crucial areas in modern psychology, how genetics and the environment combine to shape mental health. Gone are the days of asking, is it nature or nurture? Modern science shows us that it's always both, working in an intricate, dynamic partnership. In this episode, we'll break down how genetic predispositions interact with life experiences, why no single gene can determine your fate, and how cutting-edge research is reshaping our understanding of mental illness. Get ready to uncover how your DNA and your environment dance together, crafting the complex masterpiece that is you. Section 1. Genes are not destiny. When the Human Genome Project was completed in the early 2000s, it unlocked the blueprint of human life. But one key realization emerged. Possessing certain genes doesn't seal your fate. Genes provide the potential, but environmental factors determine how and when that potential is expressed. For example, you might have a genetic vulnerability for depression, but whether that develops into a disorder depends heavily on life stress, social support, and even cultural context. This concept is known as gene-environment interaction, where the impact of a genetic variant depends on environmental exposure. As David Schenk, 2010, put it, genes are not instructions, they are options. A vivid illustration is seen in studies of serotonin transporter genes, where individuals with a particular gene variant are more likely to develop depression only if they experience significant life stress. Without that stress, the gene may remain dormant. This shows us that genes can sensitize people to the environment, but they don't act like switches that turn disorders on or off by themselves. Section 2. Understanding heritability. A commonly misunderstood term in genetics is heritability. Many assume that if a disorder has a heritability estimate of 50%, it means half of the disorder in a person is genetic and half is environmental. But that's incorrect. Heritability refers to the proportion of variation in a trait across a population that can be attributed to genetic differences. For example, ADHD has an estimated heritability of 70%. This means that, in a group of people, about 70% of the variation in ADHD symptoms is explained by genetic differences. It does not mean that an individual's ADHD is 70% genetic. Additionally, these estimates come from twin studies, adoption studies, and family studies, which allow researchers to disentangle genetic and environmental contributions by comparing individuals with varying degrees of genetic relatedness raised in different environments. Importantly, heritability is not fixed. It can change depending on the population studied and the environment. In a society where everyone has excellent nutrition, the heritability of height might be very high, because environmental differences are minimal. But in a setting with widespread malnutrition, environmental effects play a much larger role. Section 3. Behavioral genetics in depth. Behavioral genetics investigates how much genetic and environmental factors contribute to variations in behavior. It distinguishes between genotype, the full genetic makeup of an individual; phenotype, the observable traits and behaviors shaped by the interaction of genotype and environment. Consider the example of intelligence. You might inherit a genetic potential for high cognitive ability, but without educational opportunities, intellectual stimulation, and encouragement, that potential might never materialize fully. Conversely, supportive environments can help individuals overcome genetic risks. A groundbreaking study by Turkheimer and colleagues, 2003, found that socioeconomic status, SES, influences how genes express themselves. In low SES families, environmental deprivation suppressed the genetic potential for intelligence, whereas in high SES families, genetic differences became more apparent. This dynamic shows that genes can only unfold their influence in certain environmental contexts. Section 4. Molecular genetics unpacked. Molecular genetics goes beyond estimating heritability and seeks to identify the specific genes involved in complex traits. Our DNA consists of 23 pairs of chromosomes, and each chromosome contains thousands of genes. Variations in these genes, particularly single nucleotide polymorphisms, SNPs, create the genetic diversity we observe between individuals. SNPs are tiny changes in the genetic code, often involving just a single letter in the DNA sequence. While many SNPs have no effect, some can slightly increase the risk for disorders like schizophrenia or depression. Another source of variation is copy number variations, CNVs, where sections of DNA are abnormally repeated or deleted. CNVs can disrupt gene function and have been implicated in autism spectrum disorder, ADHD, and intellectual disability. To investigate these links, scientists conduct genome-wide association studies, GWAS, which analyze the genomes of thousands of individuals to find associations between specific genetic markers and mental health conditions. GWAS have revealed that most psychiatric disorders are polygenic, meaning they result from the cumulative effects of many genes, each contributing a small increase in risk. Section 5. Gene-environment interactions and the rise of epigenetics. Gene-environment interaction research explores how certain genes increase sensitivity to specific environmental factors. For example, children with a genetic predisposition to impulsivity may thrive in structured, supportive environments, but struggle in chaotic or neglectful settings. This brings us to the cutting-edge field of epigenetics. Epigenetics investigates how environmental exposures can alter gene expression without changing the underlying DNA sequence. Chemical modifications such as DNA methylation can turn genes on or off, shaping biological outcomes. Perhaps most astonishing, some epigenetic changes appear to be heritable across generations. Research on the descendants of Holocaust survivors, for example, suggests that trauma can leave epigenetic marks that affect stress responses in their children and grandchildren. This revolutionary insight expands our understanding of how history, culture, and biology intertwine to influence mental health. Section 6. Challenges and future directions. Despite the impressive advances, many challenges remain. Mental disorders are rarely linked to a single genetic variant. Instead, they arise from complex gene networks interacting with neural systems, social contexts, and life events. Additionally, the same genetic risk factors can predispose individuals to multiple disorders, blurring the boundaries between diagnostic categories. Moreover, gene discovery alone cannot predict who will develop a mental illness. Scientists must integrate genetic data with neuroimaging, biomarkers, psychological testing, and environmental assessments to build a fuller picture of risk and resilience. Finally, ethical concerns loom large. How should genetic information be used in clinical settings? How can we prevent genetic discrimination? And how can we ensure that scientific advances benefit all populations, not just those represented in current research samples? Conclusion. Embracing complexity, empowering change. The study of genetics and environment in psychopathology has opened breathtaking new windows into the human condition. Yet, at its heart, the message is clear. No one is defined by their genes. Mental health is shaped by biology, experience, and context, and it is open to change. As we move forward in this series, we will continue to explore how this knowledge can be transformed into effective interventions, compassionate care, and a better future for those living with mental health challenges. Stay curious, stay compassionate, and as always, understanding, empowering, healing together.